Greetings, everyone, and good morning, good afternoon, or good evening, depending on where and when you're accessing this recording. I'm Paul Boettcher, an Animal Genetic Resources Officer from the Food and Agriculture Organization of the United Nations, better known as FAO, based in Rome, Italy. I'm currently based at the Joint Division of FAO with the International Atomic Energy Agency in Vienna, Austria. Now I'm going to share my screen and continue with my presentation. The topic of this conference is, of course, sheep production, but I'm going to expand this a bit and talk about livestock in general, specifically about the state of global gene banking of animal genetic resources. But before starting, I'd like to recognize my colleagues who have helped contribute to this presentation, specifically Michel Bochard from France, Simke Heemstra from the Netherlands, Alessandro Stella from Italy, and other members of the IMAGE consortium. IMAGE is a large European project about which I will talk about more later in the presentation. So why do we cryoconserve animal genetic resources? Animal genetic resources are an essential part of the biological basis for world food security. More than 1 billion people rely directly on livestock for a major proportion of their livelihoods. A diverse genetic resource base is critical to eradicate world hunger. Genetic diversity of livestock allows the populations to adapt to current and future environmental constraints and changes in the market. The diversity also serves as the raw material for breeders to make genetic improvement. Unfortunately, the diversity of animal genetic resources is under threat. But the population numbers of many breeds are decreasing or unknown, and many developing countries lack the capacity for animal genetic resources management. For these reasons, international cooperation is necessary. The Food and Agriculture Organization of the UN takes a major role in the international coordination of the management of animal genetic resources. In 2007, in response to the knowledge that animal genetic resources were under threat, the member states of FAO developed and adopted the Global Plan of Action for Animal Genetic Resources. The Global Plan of Action is a policy document for management of animal genetic resources. It includes 23 strategic priorities for action that are aimed at addressing current and future challenges to the livestock sector. These 23 different priorities are assigned to one of 4 strategic priority areas, the first of which is characterization, inventory, and monitoring, the second, sustainable use and development, the third, conservation, and the fourth, policies, institutions, and capacity building, which of course underlies the implementation of the first 3 strategic priority areas. As was indicated, conservation of animal genetic resources is one of the 3 or 4 key strategic priority areas. Conservation includes 7 different strategic priorities. Number 7 is to establish national conservation policies. Number 8 is to establish or strengthen in situ conservation programs. Number 9 is to establish or strengthen ex situ conservation programs. Number 10 is to develop and implement regional and global long-term conservation strategies. And number 11 is to develop approaches and technical standards for conservation of animal genetic resources. Approaches for conservation of genetic resources can be divided in 2 different ways. Depending on how the material is stored, conservation can be either in vivo, using live animals, or in vitro, using some other sort of a genetic material. You also can assign conservation programs according to the environment in which the materials are stored. In situ refers to conservation of the animals in place, which would be on farms for livestock, obviously. Ex situ simply means that are not held in their normal place. And live animals can be held in either agricultural parks or research farms, which would likely have somewhat different environment than commercial production. And the other option, the option about which I'll talk today, is cryoconservation, or gene banking of animal genetic resources using cryopreservation, deep frozen and freezing in liquid nitrogen. Cryoconservation has a number of advantages over the other approaches. It can conserve a population in perpetuity, practically, at least within our lifetime, certainly, with a low risk of loss to disasters, if it is well planned, which would often include duplication of the collections. Another advantage is that maintenance costs tend to be low, generally just a constant source of liquid nitrogen and the staff to manage the collection. This is relative to in vivo collection, where management and maintenance of live animals can be quite costly, especially maintaining a population large enough to ensure a wide amount of genetic diversity. Some disadvantages can be seen from cryoconservation. First, because you need to establish physical infrastructure and equipment for gene banking, the initial cost may be high. That stands as a little bit of an obstacle for many developing countries. In addition, specialized skills for cryopreservation and collection of material are also required, which is again another obstacle. A population in cryoconservation has its material fixed in time and therefore has no chance to evolve and adapt to populations unless resampling is done. And the sampling for cryobank collections must be significantly large and well planned to avoid populations associated with drift, selection of just a small group of founders in your collection. The strategic development goals recognize the importance of cryoconservation. I assume that most of you, if not all, are familiar with these sustainable development goals. These are officially known as the Transforming Our World, the 2030 Agenda for Sustainable Development, and they consist of 17 goals with 169 targets to measure achievement of the goals. These Sustainable Development Goals were developed through a collaborative process led by the United Nations and civil society organizations. These goals reflect all aspects of sustainable development, ranging from climate action to greater partnership to maintaining life underwater. But the most important for food production and agriculture and genetic resources is goal number 2, to end hunger, achieve food security, and improve nutrition and promote sustainable agriculture. Within these goals, there is one target, number 2.5, which deals specifically with genetic resources for food and agriculture. I'll read word for word the description of this target. By 2020, to maintain the diverse genetic diversity of seeds, cultivated plants, and farmed and domesticated animals, and their related wild species, including through soundly management diversified seed and plant banks at the national, regional, international levels, and to promote access to and fair and equitable sharing of benefits arising from the utilization of genetic resources and the associated traditional knowledge as internationally agreed. This target is measured by 2 different indicators. The first is indicator 2.51, which consists of the number of animal and plant genetic resources for food and agriculture secured in either medium or long-term conservation facilities, gene banks obviously, whereas indicator 2.5.2 is the proportion of local breeds classified as being at risk, not at risk, or unknown level of risk of extinction, obviously referring to in situ conservation and sustainable use of genetic resources. Within indicator 2.51, there's a specific unit referring specifically to animal genetic resources. This is denoted to indicated 2.5.1b, and it is the number of national breed populations with sufficient material stored in a gene bank. In this case, sufficient material is defined as the amount of material that is required to reconstitute an extinct breed to its living and viable state. FAO is a knowledge organization, and a major activity of FAO is putting information within reach to stakeholders. One of these activities that FAO undertakes under this objective is to review the implementation of the Global Plan of Action and monitor its progress every 5 years. Another example is FAO's maintenance of DADIS, the Domestic Animal Diversity Information System, which is a database on animal breeds. This figure here is a screenshot of DADIS. Within DADIS, there is data on population numbers, which is used to calculate the indicator 2.5.2, as well as cryoconservation data, which is used to monitor indicator 2.5.1b. Another example of information management by FAO is frequent surveys with stakeholders on targeted topics regarding animal genetic resources management. Within the recent years, FAO undertook with colleagues and collaborators a gap analysis of material stored in gene banks, as well as an analysis of and survey of quality management undertaken by gene banks. Looking at the status of current gene banking activities, this figure shows the number of countries having and reporting gene banks. As you can see, since the Global Plan of Action was adopted in 2007, there has been a near doubling of gene banks, from 55 to 107 countries reporting gene banks. This 107, considering that there are around 190 FAO member countries, indicates that more than half of countries now report animal gene banks to some extent. This is not necessarily a formal national gene bank, but also can include collaboration with AI centers or with universities or other research institutes. This map here shows the global distribution of animal genetic resources gene banks. As you can see, nearly all the countries in the Americas have gene banks, as well as in Europe. Within the rest of the regions, the number of gene banks is a bit lower, but there still is a very high number of gene banks within Southeast Asia and Oceania. This chart shows the number of countries within each region, or the percentage of countries within each region, in more detail. As you can conclude, the cross-region distribution tends to associate with economic development. Gene banks are more common in Europe and North America, 100% in North America and nearly 80% in Europe, whereas less frequent in the Near East and Africa. Within region, there is also a link with economic development, but there's an association with the importance of livestock. You can't see this on this graph, but for example, there are few gene banks in the island states, which tend to have small populations of livestock and do a lot of importation of their livestock-based foods. As far as species, this graph shows the percentage of countries reporting different types of species in their gene banks. As you can see, the so-called Big Five species, chicken, pig, sheep, goat, and cattle, are among the most commonly cryoconserved species, with the exception of horse being recorded as being cryoconserved as a higher level than with chickens. With regard to species, semen is by far the most commonly preserved material, which is not surprising because it is the most easily conserved material, and because ruminants for which these techniques are well developed are the most commonly cryoconserved species. Nearly 100% of countries conserve semen in their gene banks. A bit more than 40% report embryos, and less than 20% report somatic cells, oocytes, gonads, or primordial germ cells. Almost 40% of the countries report complementing their storage of other materials with storage of DNA samples for the animals with material. Evaluating the use of species and according to the use of material according to species, different strategies are taken according to species. Semen is the least invasive material, and therefore it is the most common and most important tool for cryobanking. On the other hand, there are still needs for improvement of techniques for semen and embryos, which differ according to species. For semen, chicken and sheep cryoconservation for semen lags behind that of cattle, for example. For embryos, pigs and horses also lag behind some of the other species, and also for chickens and other avian species, conservation of embryos is practically impossible. Therefore, gonads and primordial germ cells are looking like new and promising technologies for birds and other species. As using different transplantations of these materials, it is possible to regenerate a new population within 2 generations. One factor to consider about species and materials is that there are various legislative and ethical issues can influence the material choice. For example, cloning is prohibited for animals used for meat production in some countries, and therefore that could limit potentially the use of somatic cells for future cloning of animals. Looking at the choice among breeds, breeds can be assigned to different categories of risk based on their population size. Considering that one of the main goals of cryoconservation is to prevent breeds from being extinct, one might think that breeds at risk would be the most highly conserved type of breed. However, as this chart shows, that is not the case. Breeds that are not at risk are conserved at a much greater rate than those not at risk. Breeds can also be defined according to their geographical status. FAO and the member countries have agreed on a system in which local breeds are defined as those which are based only in one country, whereas transboundary breeds are those based in more than one country. Among the transboundary breeds, there are regional transboundary breeds which are found in only one continental region, or international transboundary breeds which are found in more than one continent or region. You might think that local breeds would be the greatest target for national gene banks, but in fact, international and regional transboundary breeds are conserved at a greater frequency than our local breeds. This is especially the case when you consider combining the same breed population across different countries. Considering, for example, the Holstein breed, which has the opportunity to be conserved in many countries, international breeds are conserved at a much, much greater rate than our local breeds. This distribution of conservation might seem counterintuitive, as it tends to favor the not at-risk breeds and those which are international transboundary, and therefore quite easy to be found and not at great risk for extinction. But why is this happening when it's somewhat counterintuitive? Well, there are some explanations. First, from a purely practical standpoint, these animals being more common would be easier to access to preserved material. Another explanation is that maybe the goals of cryoconservation are not only to reconstitute extinct breeds, but to be used in managing the in situ populations. And in fact, the results of another survey undertaken tend to back up that second proposal. Well, if you look at the 4 most common reasons for gene banking, 31% of countries indicate that preservation of breeds to prevent their extinction is their most important priority. Second, but very close behind, is for management of in vivo populations. As you can see, almost the same number of countries conserve for in vivo management as their primary goal as they do for prevention of extinction. Another 28% of countries, almost the same number, preserves their genetic resources in gene banks for the primary goal of research studies, whereas only 11% have the primary goal of supporting crossbreeding or integration programs. So therefore, as you can see, management of in vivo populations as a goal for gene banking is more and more becoming an important priority. Innovations are essential animal genetic resource to account for changes in these goals, as well as changes in the continuing development of gene banking technologies. In addition to this increased utilization of gene bank material for research or for management of in vivo populations, we're also looking at increased engagement with a greater group of stakeholders. The European Union has seemingly recognized this fact and recently sponsored a research project called IMAGE for short, or Innovative Management of Animal Genetic Resources. FAO was one of the partners in this project, along with 28 or 27 other institutions around Europe and the world. This map here shows the countries that contributed and had institution members as partners of IMAGE, and included not only European countries, but also Colombia and Argentina from South America, and Egypt and Morocco from Africa. The objectives of IMAGE were to increase and enhance the use of genetic resource collections, to upgrade the management of animal genetic resource banks, and to demonstrate the benefits brought on by gene banks, not only for minimizing genetic accidents like the extinction of a population or extreme genetic drift, but also to allow the livestock sector to respond to new environment constraints and new market needs, i.e. to better manage in vivo populations. IMAGE included 7 different work streams. The first was to involve the stakeholders for management, of course, of in vivo populations to a greater degree, right from the beginning, both of the project and of conservation programs. The second is to improve the management and operation of gene banks. The third is on a biological basis, looking to improve the reproductive quality of the gene bank samples and their usability. Genomic characterization of genetic diversity within gene banks. Facilitation of access to information. Developing and testing new strategies and scenarios for use of gene bank materials. And to enhance synergy between gene banks and management of genetic resources in situ. And for increasing awareness about the value of gene banks. As you can see, all these objectives really aim at improving the utilization in vivo. I'll just go over briefly some key results of the IMAGE project. First of all, I'll look at the scenario that had been used in the past for countries for which restoration of extinct populations were the main goal. In this case, the process was quite simple. You merely had to identify the breeds at greatest risk of extinction, looking at their population size, and determine the number of donors and quantities of materials. There had already been guidelines available for the number and quantity of donors in material based on the species and the type of material to be preserved. Then the final step was just to collect and store sufficient material to reconstitute the breed. And there you have it. This is a bit simplified, of course, because things change over time. You may have to repeat this process occasionally if other breeds become at risk for extinction, or also to prevent impacts of genetic drift. The new normal is slightly a bit more complicated. With utilization for more reasons, you have to consider your gene bank objective to update your collections, monitor the existing trends, and also as well to provide insurance for your breeds. In this scenario, you can look at 4 different groups of material. The first are the genetic resources held on farms in situ, and the second are 2 sub-collections of the gene bank. C is the traditional one, which would be used, if necessary, to restore an extinct population in vivo, whereas the new sort of collection is used to help enhance management in vivo population, either to restore lost variants, for example, or to increase variability within the population. This collection of B, large enough, could also be used for other alternative objectives, such as to establishing a new breed, either using in vivo animals from a different breed, or even 2 different collections from 2 different breeds held in vitro within the gene bank. This new normal requires a different and more complicated rationalization framework. Before it was a 3-step approach. Now there are many different factors considered. In the end, you want to develop your gene bank portfolio by asking yourself why you're conserving the breed. That will impact a number of other factors, including what materials to store, which breeds to store, and how much material to store. The purpose of the program will also have an impact, as well as the users. Considering this complicated aspect, you also have to now look at cost effective, because you're looking at the impacts in the in vivo population. You look at equity, because there are more stakeholders, and there also is a governance to help achieve this equity. The technical efficiency may be also greater, because first you need this cost effectiveness, and then you also may be looking at different types of materials for different breeds, different species, and different goals. You may often look at evaluating your genetic analysis over time, looking at using new genomic approaches to do the analysis of genetic diversity, and over time continually optimizing your collection. And you also need to follow with a continual access and discussion with the stakeholder to ensure that your collections are meeting their present and future needs. And this whole process is not just done one time, but is a continuing process being reviewed every few years. As I mentioned on the previous slide, we now have the opportunity to use genomic characterization to manage our gene bank collection. Under the old scenario, a lot of times the number of the pedigrees and the number of animals needed, assuming unrelated animals, were the main guidelines for targeted collection. But now we can use genomics to look at the aspects of the material within the gene bank, the diversity that's existing, and relate it to the diversity within the population based on genomic testing, and target specific animals which add new diversity to the collection. To help facilitate this process and to save cost, the AMACH project, as one of the deliverables, developed a multi-species SNP chip for evaluating both genomic collections and in vivo populations. This chip can hold up to 10 different species with 10,000 single nucleotide polymorphisms for each species, with a low cost of less than $20 or euro per assay. The selection of these SNPs was a very deliberate process, considering different types of SNP. For each species, these panels include 8,000 different neutral or different SNPs designed to evaluate the neutral genetic diversity to capture as much of genetic variability as possible. It also includes 7 different SNPs which have been associated with functional genomic regions within the genome, those known to affect phenotypic traits. Additional 300 were SNP that can use in traceabilities to ensure that the animals that are being collected cleanly and purely represent the breed from which they're intended to originate. And there also includes 1,000 different ancestral SNPs to monitor the proportion of original founders of the breed contributing to the genetic collection. All of these 10,000 SNPs were chosen from available marker sets to ensure connectivity with previous studies on genomic characterization. Another key deliverable of the project was the IMAGE data portal. With gene banking, the data regarding stored material are a fundamental concept of cryoconservation. For stakeholders to efficiently use the stored material, information about the samples must be made publicly available. Therefore, IMAGE developed a public data portal for gene banks of IMAGE partners. However, this data portal is now available for use by any gene bank around the world for dissemination of its information to its potential stakeholders. This data portal has advantages both for gene banks and for users. The beneficial features of gene banks are the following. The data portal uses well-defined metadata rules to assign each sample to a unique identifier to each donor animal of the material. There's a simple inject to allow it for either fast upload or direct downloading, transfer of data from existing databases to the data portal. One problem with gene banking or any databases in general is that sustainability over time, especially when they're associated with the project. The IMAGE data portal has ensured sustainability because it is being hosted by the European Bioinformatics Institute platform, which is a long-term funded agency. This hosting with the EBI also allows for integration with other databases, including those that provide geographic information systems, with data of FAO, and with other archives hosted at EBI. With regard to the users, this data portal can serve for a single port of entry for querying the contents of multiple gene banks. It also allows for seamless access to complementary information on gene bank samples, such as tissue type, the breed data, information about the geographic origin of the samples, genomics data if available, and phenotypic information if it had been uploaded by the gene bank. This slide simply shows the screenshot of the data portal, but especially note the URL for accessing the portal, which also includes manuals and help pages for its utilization. In addition to these major deliverables, IMAGE, of course, yielded several other deliverables, some of those which I'll mention here. IMAGE included studies on the ethics of breed selection and use of biotechnologies within gene banking. It developed novel markers for testing the quality of chicken semen and pig embryos, developed new cryoablation protocol for primordial germ cells of poultry, undertook genomic analyses of important animals in key livestock populations to improve the efficiency of gene banking, developed new methods and software associated with these methods for genetic diversity management and for integration of populations using gene bank materials. And it also held 7 different training courses for young scientists to ensure the results of IMAGE can be used for the future. For information on IMAGE and for just follow-up data on the various results, I present here the URL. I'll leave again for a second or two, although I assume you can access the recording of this presentation at a later time. So in conclusion, gene banking is an important management tool for animal genetic resources. In the past, prevention of extinction was recognized as the most important goal, but more and more gene banks are being used to complement the management of in vivo population and to provide material for research. Currently, more than 100 different countries report having gene banks, showing the importance of having such an infrastructure. Ruminants and semen are the most commonly conserved species in materials, largely due to their value and availability. Note that risk transbounded breeds are most commonly conserved type of breed. This again seems to substantiate the great utilization for management of populations within vivo. International collaboration plays a key role in enhancing the role of cryoconservation, and this will likely continue in the future. Although 100 countries already have gene banks, that still leaves nearly 100 which do not, and the management of some of these 100 countries certainly could be improved through international collaboration. I invite all of you to do this with your collaborators. So with that, I'd like to thank you for your attention, and I will now proceed to the discussion section.